Hey everybody, it's me. Um, Let me get this open. It's Sunday. I've had a good day. I've already walked a bunch. Um, why? Okay, there it goes. I painted my new front door. You'll see it on Instagram. It looks so good. I've been trying to keep busy. Um, but we're going through the last thing. We've done replication. We've done transcription, and now we're going to do translation. We have to have a test this week. I'm planning on Friday. We will do it on Plus Portal, and I'll try to make it as close to our normal tests as we've had so that it's very familiar with you guys, just like our notes are kind of the same. So let me go over translation with you guys. So, first of all, translation is basically going from RNA to make a protein. And remember, to make a protein, the monomers, which are one, has to be the amino acids, and then many amino acids make up a protein, which is the polymer. So that's what we're doing. We're getting together amino acids to make a protein here from messenger RNA. So translation converts messenger RNA messages into polypeptides. Okay, poly means many. A peptide is a bond between one amino acid and another amino acid. So, what this means, polypeptide, is there are many amino acids bonded together to make a protein. Okay? So, a codon is a sequence of three nucleotides that code for an amino acid. This right here is a codon, AUG. Okay? This is methionine. This is the start codon. Okay? AUG is the start codon. Now, this is another codon, CUU, and this is for the leucine. Okay? The genetic code matches each codon to its amino acid or function. So, this is a codon, and this is basically right here messenger RNA. Amino acid codons are on messenger RNA. Okay, this right here is a chart for amino acids. So they make this kind of complicated, but what they do is that if your first letter of your base, let's say it's, well, they're using over here CAU, okay? So if you look over here, find the base C. So you look here, this is the first base, so C. So it's going to be in these two, these right here. And then the next one is A. So then you look up here, and so you go here. It's like a battleship. So C and A are right here. C and A here. And then you're looking, third base is U. So it'd be this one. CAU is histidine. Okay, this is just how you find amino acids when you're looking at your messenger RNA with your three codons. So it's actually kind of fun to do that. So I might get you a worksheet to do those. So let's see. I'll let you know after I finish this. All right. So here is your amino acid. This is your messenger RNA. And this one right here is CGA, which is arginine. This is UAC, tyrosine. That's why you have the other one that has all the names. Okay. The change in the order in which the codons are read changes the amino acid in the protein. So if we start at the first one, which is C, we have arginine, tyrosine, serine, and serine. Okay? But if we skip the first one, then we change our amino acids. Then we have asparine, thyrronine, valine. So all they're saying is that how these are, where they start, makes different amino acids, okay? Regardless of the organism, the codons code for the same amino acid. So what this means is that if CUA is always going to be arginine, UAC is always going to be tyrosine. So they're saying no matter the organism, a cat, dog, human, insect, whatever, if they've got DNA and amino acids, it's going to be the same. All right, so this is a transfer RNA, okay? So I'm going to go over this. This right here is actually the amino acid, and a transfer RNA carries the amino acid, and then this is the anticodon, okay? So the anticodon is here, and then messenger RNA goes down here. So the messenger RNA codon is GCC, and that's coding for this amino acid right here. Transfer RNA actually has the anticodon, okay? And so this one right here, CGG, is the anticodon of GCC. So the opposite of the messenger RNA, G, is C. The opposite of C is G, and the opposite of C is G. So this is the anticodon. This is the amino acid. This amino acid is codon is GCC. So this right here is just showing you. Transfer RNA has the actual amino acid up here, and it has the anticodon. Messenger RNA is going to have the codon for this amino acid. So transfer RNAs are synthesized in the nucleus and charged in the cytoplasm. Transfer RNAs contain the anticodon, the complementary sequence to the codon in the messenger RNA. So this right here at the bottom is the anticodon. Messenger RNA actually has the codon for this amino acid. Hope that makes sense. All right. Ribosomes consist of two subunits. Remember, the ER makes the nucleus. That's where the ribosomes are made, and they're on the rough ER. So ribosomes consist of two subunits. Okay, this is the large subunit, and this is the small subunit. The large subunit has 3 binding sites for the transfer RNA. The transfer RNA anticodon goes here. Okay, these 3 sites right here, and then the small subunit binds to the messenger RNA. So the transfer RNA is going to bind right here, and the anticodon is going to be here. And then the messenger RNA goes through here, and it's going to have a codon. Transfer RNA has the anticodon. And if you wait just a minute, the next one will probably show it better. Okay, but this is what it looks like in a big picture, and then I'll show you in a smaller one in just a second. Okay, so, okay, good. The translation to begin, you start the transfer RNA. For translation to begin, you start the transfer RNA, the one carrying methionine. AUG is the start codon. AUG is methionine. That is the start codon. So it binds the start codon on the messenger RNA and signals to the ribosome to assemble. Okay? So in this picture, these are actually the transfer RNAs right here. They don't really look like a T, but this is the transfer RNA. Okay, let me back up though. This is the nucleus, okay? DNA replication and transcription all happen in the nucleus. Once transcription's occurred, messenger RNA comes out of the nucleus, and this is messenger RNA here, okay? So this is going to have the start codon, AUG, and then this amino acid right here is going to have the anticodon of AUG, which would be UAC. Okay, and so here, this is what happens, and there's going to be another video to show you, and this happens in all the ribosomes, so you can see that this is like an assembly line. This is happening in this amino acid, but this is another, this is a ribosome, this is another ribosome, this is another ribosome. Okay, so the transfer RNA has the amino acids on it, and it has the anticodon. So once it like hooks up with these two right here, and it goes to this site. These are the binding sites. Okay, so when this one comes in and it moves over, these 3 amino acids will hook to this one. And then another transfer RNA will come in, and then the amino acids will go to the next one. And there's a picture about this in a minute. So for translation to begin, the start codon, the one carrying methionine, which is the start codon, binds the start codon on the messenger RNA and signals to the ribosome to assemble. Okay. The ribosomal RNA catalyzes the formation of a peptide bond between the amino acids. So when see this right here is a peptide bond. When this one right here, this is the start codon, AUG, when this one comes down, then this methionine will go to that one, and that's what the peptide bond is. Ribosome ratchets the messenger RNA down 1 codon length. Okay, so leucine's going to come in through here, and then when the next incoming transfer RNA is, this one goes here, this one goes here, and then the new one will go here. And it just keeps passing on the amino acids until you get to the start codon. Okay, so these right here are really good to read, and I'm going to show you, I think I went too far. Okay, and I'll show you a video. Okay, the empty transfer RNA is released. So this one right here is released, and the new complementary transfer RNA enters the ribosome complex. This is the new one. Okay, so this one was here, and this one was here, but then they moved down. So this one left, and now its amino acids are here. So now this one comes in, okay, And then the new complementary transfer RNA enters the ribosome complex, and once the stop codon is reached, the ribosome complex disassembles. So this keeps going until it gets to the stop codon. Okay, this one right here is the stop codon. And you can see that more amino acids or transfer RNAs came because this keeps getting bond. This is a polypeptide bond. This is a peptide 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 bond. All of these together are called polypeptide bonds. Okay, and it just keeps going until you get to the stop codon, which is right here, UAG. There's 5 stop codons. Okay, this is a really good video, but it's very long. So I'm going to show you in the next one. I also want you to, oops, hold on. Want you to look here real quick while I've got this open. All right, so this is on page 668. This starts translation. Please read this part. This one, sorry, I keep coughing. This really goes through all the things too. So go look at that too on page 668. And then if you look here, this is the sites that we were showing you. Transcription happens through the nucleus, and then messenger RNA comes throughout, and it's got all these little things, and messenger RNA comes up here, and this is the enzyme that's carrying the transfer RNA with the amino acid here, and you can see that this is a polypeptide bond, and this is the new one, and then it's going to have the yellow one, and you can see it going through all of its little things. This one right here, this video is the best video. Okay, so again, I'm going to let you watch this. This is on 670. It is the transcription. It's a great video. It's going to show you exactly what everything's happened. So you're going to need to look at this page right here. So start at 668, okay, right here. Look at this. This one has a whole bunch of buttons you can push, okay? And then go to this one, read that. Sorry. And then play with this one because you can go through all the little buttons. And then watch this movie. It's really good. Okay? Watch this movie. Bye, y'all.